Thank you, Esther, and good afternoon. I'm Debra Niehoff, AFTD's Director of Research and Grants. For our next session, we're going to talk about biomarkers. What are they? What do they have to do with FTD? And how you can get involved in important biomarker-focused research. And we do have a slide. Thank you. AFTD has recognized the importance of biomarkers and invested in biomarker research for over a decade. And admittedly, at first, we did this on a small scale, giving modest grants in some of our longest running programs, like our pilot grant program. We took a big step forward in 2016, thanks to a generous donation from the Samuel I. Niehoff Foundation that allowed us to launch the FTD Biomarkers Initiative, our first program devoted exclusively to funding biomarker research, and we funded 13 grants in the US and Europe for research on biomarkers. And another step forward was when we joined up with our longtime partners, the Alzheimer's Drug Discovery Foundation, Bill Gates, and other funders, to participate in the Diagnostics Accelerator, to take promising biomarkers one step closer to the clinic. And we currently have 4 active awards in that program. To learn more about biomarkers and biomarker research, we'll be speaking with 2 experts in the FTD field. Dr. David Irwin is principal investigator at the Penn Digital Neuropathology Lab and the attending cognitive neurologist at the University of Pennsylvania's Frontotemporal Degeneration Center. Dr. Tania Gendron is assistant professor of neuroscience at the Mayo Clinic in Jacksonville, Florida, where she leads research conducted by the Translational Neurobiology of ALS and FTD Laboratory. Welcome to you both.
Thanks so much, Debra, and thank you to the AFTD and to all of you to have the opportunity to speak today. I'll be talking about observational research in frontotemporal degeneration, and we'll bring up the slides, please. Yes. Would you like, I think we may be out of order. Yes, okay, thank you. So we'll be talking about biomarkers and research in FTD. Next slide, please. So starting the talk, we'll be talking about the diagnostic problem or the difficulties we encounter in clinic, which is the inability to diagnose patients with the underlying cause of FTD. This is important because the biology of FTD is what guides eventual treatments, and something analogous is cancer research, where a biopsy of liver or lung is fairly easy to obtain, and a pathologist can go right under the microscope and say the exact cause of the disease, and then tailor a treatment for this. So therapeutic trials are coming for FTD and are here, and we're excited. And to expand this to as many patients as possible, we need better tests or biomarkers. And that's the solution part of the talk that we'll be going over. And a big part of that is observational research. I'll go through the definitions of different types of research and what's involved, and specific questions that you may want to ask your doctor when considering participating in research. And particularly, the importance of brain donation is also a very important part for us to help understand and accelerate the science of FTD research. So as you know, FTD is caused by several different diseases under the microscope, and we have clinical symptoms that we use to construct and help with the diagnosis. Many of these are things that are hard to measure, like social behavior, language, and can take time to notice a change over time that can help us with the diagnosis. And even doing an imaging study of the brain, such as an MRI, as you see here, can show us the parts of the brain that are having difficulty. But even if we obtain a brain sample to look at, the actual cause of the disease isn't clear until we go under the microscope. And there are two main types of what we call proteinopathies, or biological causes of FTD. One caused by the tau protein, the other a very different protein called TDP43. And these are examples of what it looks like under the microscope, and they're very different. And the biology of these proteins are also very different. Tau helps stabilize the brain cells and keeps them communicating with each other, whereas TDP43 is found in the control center of the cell, which we call the nucleus, and it helps the cell regulate gene expression and respond to different challenges during life. So therapies that would target these proteins and the associated process with the disease would likely be very different. So as we need to be able to detect these proteins or pathologies when we see a patient. And we heard from Angela Taylor earlier this morning about nomenclature and the nomenclature, and I'm sure that many of you have had challenges with different definitions by different doctors. And this is an example of how clinicians who see patients have one terminology called frontotemporal dementia, and the neuropathologists that ultimately diagnose it definitively under the microscope have a slightly different nomenclature called frontotemporal lobar degeneration. And more specifically, the different subtypes, tau, TDP43, something called FUS, and others. So to be able to take down the wall between these two disciplines in medicine, clinicians, neurologists, psychiatrists, and neuropathologists, we need tests to link them together, to link the clinical syndrome of FTD with the actual biology that's causing the disease. And we do this with biomarkers that we'll hear about today. Why is this important, and how do these proteins and the associated processes cause the symptoms that we see in patients? One potential analogy is a pop-up ad. I think all of us have had an experience in life where we've clicked what seemed like a regular button on the website, and it's caused a cascade of windows that have taken up the whole screen, and the computer is slowed down and hard to do other functions. As these proteins that we all have in our brains that do normal functions for reasons that we don't fully understand, they can start to stick together and aggregate and possibly spread from cell to cell and slow down normal neuronal processes. So that's why it's an important target for therapeutics. So here you can see the microscopic pathologies and some of the different syndromes that are associated with FTD. And to have effective therapies, we want to target these proteins, their associated biology, so we can stop the development and progression of symptoms. So to summarize, there's multiple different pathologies or microscopic causes of disease that result in FTD. We think it's likely years that these processes go on a microscopic level before patients show symptoms. And that gives us a window of opportunity to be able to intervene if we can detect them. Right now, we lack the clinical tests or biomarkers that are used to detect these specific pathologies. So that's why we urgently need tests that we can use in clinic and in clinical trials to be able to say the specific microscopic cause of the disease while we're seeing a patient. And this will help us develop therapies. So moving on now to the solution. A major part of the solution is research and helping us understand the biology and why these different proteins cause different forms of FTD. So a few definitions that are useful. Research is any type of study done to answer a question. Research is done with a very deliberate cause. We want to help answer questions that are clinically meaningful, that will help change our practice in clinic. And clinical research involves any studies where people participate. This could be patients or healthy volunteers. There's other associated terms that include a clinical trial, protocol, study, or experiment. Clinical research can be further divided into two main categories, one called observational research or natural history studies, and the other is clinical or therapeutic trials. And this can be confusing. The main difference between the two studies, they involve patient and human participation. They're used to ask a question and answer a question, but the main difference is that a clinical or therapeutic trial has an intervention. It's testing an intervention on a clinical outcome. Whereas observational research, there's no intervention. It's collecting data, samples, and both are very important and both are very tied together in an important way to help advance science. Observational research can be further divided into the types of data or samples that are collected. And this could be done in one visit or one time point, which we call cross-sectional, or it could be longitudinal over time. And it could take all shapes and forms. It could include questionnaires, paper and pencil tests to help develop new tests that are sensitive to the features of FTD with language, behavior, and other motor features, for example. Imaging studies is a picture of an MRI here that you can see to look at the structure of the brain, genetics and understanding those risk factors and other genetic features that may help influence the disease, as well as collecting fluids, blood tests, spinal fluid. These are important ways for us to understand the biology of FTD, and these directly inform how clinical or therapeutic trials are conducted. And they help develop new biomarkers that can make those trials more likely to succeed. So clinical therapeutic trials can be further subdivided by the type of intervention. We often think of pharmacologic clinical trials where we're testing a drug or a medicine. That could be further divided by the type of medicine. It could be a medicine that's helping with symptoms, and that could be with sleep, mood, behavior, or what we call disease modifying. Those are types of interventions that are targeting the underlying biology, like the proteins tau and TDP43 and the associated microscopic disease functions. Then there could be non-pharmacologic interventions. These include devices or even behavioral interventions or caregiving coaching programs, things to help give information or structure. So research takes all shapes and forms, and as a potential participant, it's important to ask questions to understand what would be involved and what you would like to do for you. So to briefly mention lumbar punctures in particular, the old term for this was spinal tap, which has a negative connotation, but a lumbar puncture really is a straightforward bedside procedure that doesn't require any anesthesia. It requires local anesthesia, similar to what you would get in the dentist. And the whole procedure takes about 30 to 45 minutes. And why that fluid is so important for research is because the brain has a barrier. When we look at the blood, it doesn't always tell us what's happening in the brain. The spinal fluid is as close to the brain as we can get under the microscope, and it's relatively non-invasive. It's right in the lower back. So this helps us overcome that blood-brain barrier. For clinical use, sometimes FTD can be hard to diagnose early on, and other non-FTD diseases that involve inflammation of the brain can look just like it. So sometimes we use this as a clinical test, but it's also important for research and critical for us to make new biomarkers. And also helps us develop biomarkers in blood. If we can look at a paired sample in spinal fluid and blood, that helps us tremendously. And we've seen this in the field of Alzheimer's disease, where in the research realm, there are very established assays or tests that can detect plaques and tangles with fairly high sensitivity and specificity. And now we need those tests for the tau and TDP43 and FTD. And again, this approach can also be used in a therapeutic trial that's targeting disease, so we can track for what's called target engagement. Does the intervention in the therapeutic trial targeting the biology that we see under the microscope respond? Can we measure something in the spinal fluid that shows that there's a response? That helps the trial be more likely to succeed. So this is a really important part of research. Another very important part and sensitive issue is brain donation. Until we have a cure for these diseases, brain donation and having an appropriate test where we can detect the microscopic pathology in a living patient, microscopic analysis of the brain tissue directly is the only definitive diagnosis. And that information is so critical. All the information that we collect for research during life, having the diagnosis of what the specific pathology, whether it's tau, TDP43, or the other associated proteins, not only validates those data and fluids that were collected during life, it also tells us where to look for new biomarkers. So by looking at the brain tissue directly, we can help make new biomarkers that are specific for tau and TDP43. So brain tissue is critical for discoveries of new biomarkers and therapeutic targets as well. At University of Pennsylvania, we provide a formal report from the neuropathologist, and our clinicians review with families. So not only is it for research and the tissue is used throughout the world to advance discoveries, but also we provide counseling and support of the final diagnosis of the answer of what a patient had during life. This is an example of observational research where brain tissue and brain donation helps accelerate discoveries. This is a high-resolution MRI. The details aren't important, but the idea is that the MRI takes a picture of the brain. We have clinical MRIs, but they don't have enough sensitivity or resolution to see tau and TDP-43. And in this study where we had brain donation tissue, we're able to put it in an MRI and take a very high-powered picture that was almost like looking under the microscope. And then we could actually look at the tissue under the microscope and pair them together. And we saw very different patterns in that microscopic scale. And we're working now to take that microscopic scale and that high-resolution picture to be able to do a virtual biopsy in a living patient. So hopefully this helps illustrate how important brain donation is to accelerate discoveries. We're active in observational research at Penn. Dr. Murray Grossman, who's the founding director of the Penn FTD Center, has been doing observational research for several decades, and we're also an active site in the All FTD Consortium, which and the For Repeat Tauopathy Neuroimaging Initiative and the National the Neurofilament Surveillance Project. These are very important NIH-funded collaborative efforts. We're all working together in the scientific community to help develop new biomarkers and tests that ultimately will inform new treatments. And here is a list of the different types of activities we have, including biofluids, imaging of the brain, different types of analysis of speech, language, cognition, as well as brain donation and ways to image the eye that may tell us about these proteins too. We're also active in therapeutic clinical trials at Penn, as many sites are, and we're excited about the opportunities to be able to test new therapies and work to develop new treatments. I highlight here, we have a symptomatic therapy testing for improvements in language with a small amount of what's called transcranial stimulation, which our colleague Roy Hamilton leads, as well as a phase 3 study on immunotherapy targeting progranulin mechanisms in FTD patients and at-risk family members. And finally, we're active in gene therapy trials for FTD as well, and we have several trials for different forms of familial and sporadic FTD with the progressive supranuclear palsy syndrome, as well as the semantic variant of PPA led by UCSF. So the two types of research are really important, and they inform each other. So observational research collects data to develop new tests and biomarkers. Clinical therapeutic trials test interventions on clinical outcomes to try to slow or stop the disease or help with symptoms. Biomarkers are experimental tests that help us improve the diagnosis of these microscopic diseases that right now in clinic are invisible to us. And our goal is to help get that tissue diagnosis or the actual biological cause of the disease so we can treat it. And brain donation is a critical aspect of observational research. And this helps us develop new targets for therapies as well as help validate or make new biomarkers to improve diagnosis. So I'll end here with some resources that I hope are helpful, including our website. This fall, we're going to have our patient and family conference, and we have videos of our previous conference archived on our website that I hope are helpful too. So thank you so much for having me here today, and I'm really honored to be part of the session this afternoon. Thank you.
Hello, everyone. I'd like to thank the organizers for giving me the opportunity to share a broad overview of biomarkers and FTD research. Let's see. So there are multiple types of biomarkers, and they come in different forms. The more traditional types are proteins or other molecules that you can measure in spinal fluid or blood. There's also looking at changes in brain atrophy or white matter changes from brain scans. And newly emerging biomarkers include digital biomarkers, and these are showing promise in really providing a comprehensive data set going forward. And we'll talk a little bit about these digital biomarkers a bit more later in the talk. So just as there are different types of biomarkers, biomarkers can have several utilities for FTD. For instance, they can be used in almost all stages of drug development. So very early on, they can be used to screen hundreds to thousands of drug candidates using very simple preclinical models of FTD. When there are some promising candidates, we can then use the biomarkers to test the efficacy of these candidates in more complex models, like mouse models of FTD. And they do play a really large role in multiple aspects of clinical trial design and in the interpretation of the data coming out of the clinical trials. There's also several other important functions of biomarkers, as mentioned earlier. For instance, one of the important goals of biomarker research is to identify biomarkers that will allow us to differentiate frontal temporal dementia from other neurological diseases that sometimes have mimicking symptoms so that we could have a more rapid diagnosis. So some of you are quite familiar that there's a delay typically in diagnosing FTD. So we do hope to improve our ability to diagnose FTD in a rapid form, and this would also allow individuals to participate in clinical trials sooner in their disease course, where we believe a treatment is likely to have the most benefit. However, at present, no one test or biomarker can presently diagnose FTD with certainty. We also mentioned that it's very important to differentiate the type of FTD. So is it an FTD caused by tau pathology versus an FTD caused by TDP43 pathology? As mentioned, at present, we can only distinguish these cases post-mortem, so if there's an autopsy performed, but we can't say with certainty presently whether someone has FTD tau or FTD TDP prior to death. But it's very important that we figure this out because, as mentioned, there are therapies that are being developed specifically for FTD tau and those specifically for FTD TDP. And when those therapies become ready for clinical trial, we want to make sure the correct individuals are enrolled in those studies. So while we are making progress addressing these two important questions, perhaps some of the reasons why we have not solved them is based on the approaches we've been using. So far, we've been really using hypothesis-driven selection of the biomarkers we wish to test. What this means is we look at the research that we're conducting, trying to find the molecular mechanisms that are causing FTD. And we use this information to test what we think are likely biomarkers. And while we know a lot about what causes FTD at the molecular level, there's still so much we do not know. There's this black box, if you will. But with new advances in technology and in bioinformatics, we're now better poised to take an unbiased approach. So for instance, rather than looking at a panel of our favorite proteins, if you will, we can now measure all detectable proteins in spinal fluid or other metabolites or molecules. And then we can compare those proteins or molecules that are different in individuals with FTD to the molecules that we find in someone who is clinically healthy. We can then use these really large data sets that comprise all of this data, combine it with clinical information and demographic information, and with machine learning, hopefully come up with a panel of traits and biomarkers that will help answer the key questions that we have. For instance, can we expedite the diagnosis of FTD compared to other neurological diseases? Can we distinguish FTD tau from FTD TDP? And another important question is, can we indicate which patients are likely to have a slowly progressive disease versus those that will have a very rapidly progressing disease? And this is information that's important to the patient and their family members and their caregivers, but it's also very important in the design of a clinical trial. So imagine if you have a disproportionate amount of individuals that have a rapidly progressing disease versus a slow progressing disease in the placebo versus the treatment arms of that clinical trial. It does make deciphering whether a therapeutic had a beneficial effect much more difficult if both groups aren't proportional. Another goal that is very important to us right now in the field is trying to find biomarkers that will help us predict when someone who is presently asymptomatic but who has an FTD causing mutation will become symptomatic. And the reason for this is because if we knew when they're about to phenoconvert, so go from the asymptomatic to the symptomatic phase, then we could start enrolling these individuals in prevention trials or early treatment trials. Without a biomarker, we would have nothing to measure within these clinical trials, and so their inclusion would be nearly impossible. And so we are making some encouraging steps towards this. In fact, one protein measurable in CSF, in spinal fluid, and in blood is showing great promise, and this is a protein called neurofilament light. And we also have some imaging data that is also very promising. But as alluded to before, maybe one biomarker is not sufficient. Perhaps we do need to include biomarkers and clinical data to really get a better picture of the landscape. So I just wanted to bring up this one slide of data that this study was led by Drs. Adam Staffaroni and Dr. Adam Boxer. And what they did was for FTD caused by a C9 progranulin or MAPT mutation, they looked at different clinical traits, imaging traits, and this protein called neurofilament light. And on the x-axis of the graphs, at the time point 0, that's the time at when someone with the mutation converted, and everything before that time was while they were pre-symptomatic. And what they essentially found was that the levels of neurofilament in blood and changes in their brain started way before the time of phenoconversion, where first symptoms occurred. So these are at least two traits that we could follow over time to kind of deduce when someone may be coming or approaching phenoconversion. I'd like to spend just a little bit of time talking about digital biomarkers. These are relatively new to the field, if you will, and they are biomarkers that are quantifiable, physiological, and behavioral measures that are collected by digital devices. So this could be your smartphone, a wearable like a smartwatch, and they can even be implantable devices or even digestible devices. And the health outcome predictions are made by two ways. So there's collection of active data where the individual has to enter a value or complete a test on one of these devices, for instance, completing a cognitive test on their phone. But there's also acquisition of passive data. This is data that is automatically collected. You don't have to put any effort in. So imagine your smartwatch tracking your heart rate or your oxygen saturation, if you will. So the advantages of digital biomarkers is that they're less costly and less invasive. They do provide a rich longitudinal data set, so it repeatedly collects data over time, every day, month, year. And then we can use this rich data set to mine it for disease signature. So how does the data set from an individual with FTD compare to the data set that we capture from someone who is healthy or control? There is also a very low patient burden for these types of digital biomarkers. Again, spinal taps are relatively easy, but they are comparably more invasive than wearing a smartwatch and collecting passive data, for instance. There are numerous traits that can be collected from these devices. It could be looking at the speed of your gait, stride length, eye movements, changes in your speech and language traits, which would be relevant for PPA, executive function, which is something that's impaired in patients that have FTD, and so on and so forth. So there really is quite a lot of data that can be collected from these measures. And so here I'm just showing you one example of digital data that is now being collected. This is from the AFTD mobile app, and it was developed with the Data Cubed Health, and essentially provides the patient-reported outcomes. So individuals
would complete cognitive, motor, and language tasks on their iPhone or their smartphone using the app, and the app would also collect passive data, including the screen time and their steps, for instance. The participants have been receiving gift codes from Amazon through the app for completing the task, I think in a sense to keep them engaged and willing to do it. And based on the screenshot of the app, to me it looks like it's gamified to some degree, which also I'm sure helps with involvement. This is still in the preliminary stages, but so far the data do suggest that the tests are reliable and valid. And so combining these digital biomarkers with our fluid and our imaging biomarkers and with a rich clinical data set that also includes neuropsychological tests, for instance, really, I think, will push us closer towards effective biomarkers that can improve diagnosis, that could facilitate the enrollment of individuals early in the disease course. That will be useful for looking at endpoints and how effective treatments are in clinical trials, and will inform prognosis. So overall, biomarkers can certainly improve patient care through various ways. We had talked earlier about the importance of patient participation in research and clinical trials, and I have to say, as someone who does biomarker research, it could not happen with the participation of individuals with FTD and their loved ones donating samples, be it blood, spinal fluid, hopefully eventually digital data. And again, brain donations are incredibly important. As mentioned, we cannot know the diagnosis until an autopsy is performed by a neuropathologist. For those of you who have participated in such studies, thank you so much, and I urge you to continue. And if you are looking to participate, I list here multiple different institutes and programs that do biomarker research and collections of fluids. So please consider participating. And this is not only for individuals who have FTD. We really do need samples from individuals who do not, so that we have a comparison group. So I open this up to everybody in this room. So thank you very much.
Thank you both. We have some time now for questions from both those of you who are in our virtual audience and also everyone here in the room. And while we're waiting for questions to come in, I thought I might start. Dr. Gendron, you mentioned a protein called neurofilament light, and that is a very exciting protein in the biomarker world right now. You also mentioned the Neurofilament Surveillance Project, and that's one of the grants that AFTD is helping to fund through the Diagnostics Accelerator. So I wondered if you'd tell us a little bit more about that.
Sure. Can you hear me? Is my mic on? Yeah. So neurofilament light is a protein that is essentially indicative of neuronal injury. So initially, it will, when the cells are injured, it makes its way into the spinal fluid and eventually into the blood. So what we have found, and many of our collaborators and colleagues in the FTD field, is that when neurofilament levels are high, you have more severe symptoms. For instance, in individuals with FTD, and this is true for all FTD syndromes, be it behavioral variant FTD or primary progressive aphasias or CBS or the others. What we've also found is that, as mentioned in the talk, for those individuals who have an FTD-causing mutation but aren't yet symptomatic, it does begin to rise prior to symptom onset. And we hope that that will help us determine at what point individuals will become symptomatic. And in this way, neurofilament serving as a susceptibility or risk biomarker. Whereas in patients with FTD, we typically use it as a prognostic biomarker, trying to assess whether it can determine how quickly the disease will progress or how severe it will become. Neurofilament is also, could also be used to facilitate the diagnosis of FTD when paired with other clinical information. it's not, higher levels of neurofilament aren't indicative of FTD only. It will go up in almost all neurodegenerative diseases, but when paired with the correct clinical information, it could facilitate an earlier diagnosis. So it certainly wears many hats. It could be it could help with diagnosis. It's a prognostic biomarker, and we hope to establish firmly that it is a susceptibility or risk biomarker.
Thank you. Dr. Irwin, you mentioned the importance of brain donation to biomarker research. And we have a question here from someone in the Baltimore audience who would like to know, what steps should you take if you're interested in brain donation?
Sure, that's a great question. Brain donation access can vary depending on your geographic area and your access to different aspects of care. Most hospital systems offer autopsy for any individual seen in that health system, and that can include to get a formal diagnosis microscopically. To do brain donation for a research program, that usually requires being enrolled in a clinical research program during life and enrolling in the brain donation program, because for funding sources to be able to do the aspects of research, those are set through the funding sources for the program, but there are other opportunities. And the Mayo Clinic, for example, has a wonderful program that has outreach for brain donation, even for individuals who aren't seen at that center. So there are a lot of good resources, and perhaps I could put together some to share.
Thank you. We have a question here about brain imaging from someone in our virtual audience. What's the difference between a PET scan and an MRI scan in terms of what it shows? Is there a limit to how often people can have a PET scan? A little bit more information about brain imaging as a biomarker.
I'll defer to you.
Okay. Sure. So imaging, MRI is usually a measure of structure of the brain. So it takes a picture of the brain that could tell about the structure in different forms. A PET scan uses nuclear medicine technology and uses a radio tracer, which is essentially is an injection, and there's usually a time interval to wait for that to circulate to the brain. And then the most common clinical PET scan is something called FTD PET, and that looks at glucose metabolism in the brain. So sometimes the structure of the brain in FTD, particularly early on, we don't see signs of atrophy, but we could see perhaps that there's areas that aren't metabolizing glucose, and that could aid in the diagnosis. That involves a small dose of radiation, whereas MRIs have no radiation. It's a magnet that takes the picture. There are newer PET ligands or tracers or tests that are available largely in the research realm to start to detect the proteins of Alzheimer's disease, PET amyloid and PET tau that can bind to plaques and tangles. Those are FDA approved, but they're not covered by most insurers, so usually it's performed in a research study or out of pocket. There's intense work to try to develop new tracers that are specific for the tau that's in FTD, which is a different form of tau, which doesn't react to the other tracer very well, and tracers for TDP-43, but we don't have those at this point.
And we have a couple of questions related to participation in testing the mobile app or in some of these longitudinal studies. And I wondered if either of you would comment on what people can do if they want to be involved in testing those biomarkers, being involved in FTD.
So it's my understanding that there are multiple sites across the North America that are all FTD sites. So I urge you to go to the All FTD website to see where the closest site to you might be. And I think it is rather simple to enroll in those programs. They'll explain everything about their program that there is to know. That it typically involves the collection of fluids, I think repeatedly if you would like to do that. But if you're only comfortable giving your fluids once, we would definitely say yes to that, even if you wouldn't want to come back more than once. So they'll collect your blood, spinal fluid if you're willing, and also you'll undergo a battery of tests so he can get more clinical information associated with what might be in your blood at that time or your spinal fluid.
But they do have, as I mentioned, multiple sites. And if that's a great place to start if you'd like to enroll in a trial that will help research on FTD, including biomarker research. So again, that's ALLFTD, I think it's .org. And there's also clinical trials that have, so if you go onto the clinicaltrial.org site, there's clinical trials that are just to test digital biomarker collection and other types of clinical trials, be they observational or therapeutic, as mentioned earlier. And if you talk to your neurologists or your primary physician, they could probably also steer you in the right direction.
And finally, I just want to mention that on the AFTD website, on our featured studies page, that we do include studies that are on biomarkers as well as other types of FTD research. So that's another place where you can look to get involved in some of these studies. And I have a question. I know neither of you have a crystal ball, but I have lots of questions here, both from the folks here in person and at home. When do you think there'll be a biomarker in the clinic? One person asked specifically about neurofilament light, but the others as well. When do you think we'll have a diagnostic biomarker?
Diagnostic, I can't say with certainty, but neurofilament is, of all the biomarkers, in my opinion, the closest to making it to clinical use. In fact, I do believe that there are some clinics that are presently implementing it for FTD, ALS, and other neurological diseases. And the more studies that have been published on it so far have really provided ample data to suggest their utility in multiple ways. So I do hope, and I'm trying to push to get at least neurofilament to be more commonly used. What do you think?
Yeah, I agree with you in terms of neurofilament being a leading candidate. And I think it's an interesting or important thing to note, too, is the difference between a research and a clinical test. When we get our cholesterol checked, you can get anywhere in the world and get a number that means something. You tell your primary care doctor 111 means X, Y, and Z. With a research test, every individual lab is very, very good, but it takes an enormous amount of validation to develop an assay in one lab and have it get the exact same number in another lab. And that's kind of the main, or one of the main obstacles to get a test in clinic. But that being said, even in a clinical trial scenario, in a research realm, these things are coming close, and we're guardedly optimistic that in the next few years we'll be able to inform patients in research programs, particular about the underlying biology sooner than later.
Thank you. Looking ahead to the future again, can biomarker research tell us something about, we heard about the big 3 genes, but we know there are, and we heard about some of the rare genes, but we know there are probably genetic factors we haven't discovered yet. And can biomarker research help us there?
I do believe so. I do believe as we adopt more unbiased approaches and just try to determine what molecules differ between individuals with FTD from controls, that will uncover not only potential biomarkers, but really inform the disease mechanisms that were not really acknowledged yet. So I do think that we will learn a lot from these emerging studies that take a more comprehensive approach.
I agree. I think the ability to look at the genome is transformed in the last few years as a whole discipline called informatics, which takes these very large data sets that used to be a very difficult thing to obtain, but now we can look at the genetic code in all different ways, and discoveries are much more feasible now to help understand what are those risk factors that may alter diseases, even within the big 3. There's a lot of heterogeneity that we don't understand, and by having blood samples from families and patients who both have gene changes and those who don't, that helps, it's critical to develop and understand these new modifying factors.
Mm-hmm. Well, thank you. It was very helpful to hear all of that great information. And so thank you to Drs. Irwin and Gendron. Thank you to everybody who asked a question, both here in Baltimore and our virtual audience. It's now time. Sorry. And now it's time for another 15-minute break, and that will be followed by the afternoon breakout sessions at 2:45. For those of you who are here in person, the session on open discussion for FTD care partners will take place in Salon A through C down the hall. Building FTD-friendly communities will be held right across the hall in the Virginia and Pennsylvania rooms. And participating in FTD research, advancing FTD science, will take place right here in the Grand Ballroom. For those of you who are watching virtually, your breakout sessions will also begin at 2:45 Eastern Time. And during the break, again, we want to remind you to please consider visiting the exhibitor booths or the chat rooms during that time. Thank you.